Welcome everyone. Today we are going to learn about how lipid composition influences the physical properties of plasma membrane. Now I will talk about four important physical properties. First is membrane fluidity. Membrane fluidity. Then is thickness of the membrane. Next, curvature. And fourth is stability. Now at first I will talk about membrane fluidity, how lipid composition of the plasma membrane influence the membrane fluidity. At first I want to mention that plasma membrane is lipid bilayer in nature. The outer layer of the membrane is called exoplasmic leaflet. Exoplasmic leaflet. And the inner lipid layer is called cytosolic leaflet. Now the inner layer actually, as it faces cytosol, so it is called cytosolic leaflet, and the outer is called exoplasmic leaflet. Now it has been found, I am talking about membrane fluidity. So first point is almost all sphingomyelin and phosphatidylcholine. Now sphingomyelin and phosphatidylcholine are two lipid molecules. In my previous video, Basic Facts on Plasma Membrane, I have mentioned that in the plasma membrane there is three major categories of lipid molecules present. One is called phosphoglycerides. Another is called sphingolipids. And third is cholesterol. Now phosphatidylcholine is under the phosphoglycerides lipid molecules and sphingomyelin is under the sphingosine lipids. It has been found almost all sphingomyelin and phosphatidylcholine, these two lipid molecules, are present in the exoplasmic leaflet, that means outer leaflet of the plasma membrane. So almost all sphingomyelin and phosphatidylcholine lipids are present in the exoplasmic leaflet of the membrane. And presence of sphingomyelin and phosphatidylcholine makes the membrane less fluid. Now why? In my previous video, sphingolipids, I have mentioned that sphingolipids contain fatty acids that is longer and more saturated. Longer fatty acids can make extensive van der Waals interaction with its neighbor lipid molecules, and saturated fatty acids are straight, so it can make stable van der Waals interaction. So van der Waals interaction is important. So sphingomyelin contains long and saturated fatty acids that can make actually aggregate with the neighbor lipid molecules tightly, so it makes the membrane less fluid. Another point we can remember that falls by the lipid composition is that phosphatidylethanolamine, phosphatidylserine and phosphatidylinositol, these three lipid molecules falls under phosphoglycerides. Now they are preferentially, remember the term, preferentially present in the cytosolic leaflet. Okay, so they are present preferentially to the cytosolic leaflet, and these kinds of lipid molecules actually make the membrane more fluid. Okay, so these are two different lipid composition can influence the fluidity of the membrane. Another point you can remember, third, is high concentration of cholesterol that is present in the natural plasma membrane actually makes the membrane less fluid. Why? Because the cholesterol has a steroid ring that can intercalate with the lipid molecules that are present in the surrounding of the cholesterol, and it actually tends to immobilize the lipid molecules. Basically, they intercalate with the fatty acids, and they actually tends to immobilize these lipid molecules. So they make the high concentration of cholesterol make the lipid bilayer less fluidic in nature. By the way, the opposite will be true, that low concentration of cholesterol will make the membrane more fluid. So see how lipid compositions control the membrane fluidity. Now let's talk about thickness. As I have mentioned, plasma membrane is made up of two bilayer. Now if the two bilayer contain lipids like phosphatidylcholine, let's suppose the two bilayer contain phosphatidylcholine. So one phosphatidylcholine here, another phosphatidylcholine is here. Okay. Let's suppose these two fatty acids are present in the two bilayer, which means two lipid layer, and this is the example of phosphatidylcholine lipid molecules. Now the thickness of this molecule or these two lipids are approximately 3.5 nanometer, and in case of sphingomyelin, if the two bilayer contains sphingomyelin, then the thickness of this molecule of these two lipids will be 4.6 to 5.6 nanometer. So what I am talking about, if the two lipid layer of the plasma membrane contains phosphatidylcholine, both are phosphatidylcholine, then the thickness is 3.5 nanometer. If it is sphingomyelin, then the thickness is different. So thickness is also controlled by lipid composition. Now another point I can add, that cholesterol can increase the thickness. If cholesterol intercalates with phosphatidylcholine, then the thickness of this bilayer increases from 3.5 nanometer to 4 nanometer. Okay, remember this. So cholesterol intercalates and it can increase the thickness of the phosphatidylcholine phosphoglycerides. But if cholesterol intercalates with sphingomyelin, let's suppose in the both bilayer, in the two layer, it cannot increase the thickness of the sphingomyelin. Why? Because as I have mentioned, sphingomyelin contains fatty acids that is long, that means number of carbon atom is more in case of sphingomyelin fatty acids. I have already drawn here. And another is it is saturated, so the fatty acid is straight. Okay, so the fatty acids present in the sphingomyelin is optimally stabilized. That's why intercalation of cholesterol, so these are cholesterol, intercalation of cholesterol cannot increase the thickness of the molecule. So I can write here that 0.5 is number 0.5 is the so I can write that phosphatidylcholine, if two bilayer contains phosphatidylcholine, the thickness is low if it contains the bilayer. Remember, if the bilayer contains sphingomyelin, then thickness of the membrane is high. Now let's talk about curvature. Now what is curvature? As I have mentioned that plasma membrane is lipid bilayer, so let me draw a plasma membrane. Let's suppose this is a cell Okay, and this is the plasma membrane. Okay, this is the plasma membrane. Now if you zoom this blue line, you will find that there is two lipid bilayer actually present in this plasma membrane. So these are bilayer structure. Now in the plasma membrane and also in the intracellular organelles, I have mentioned in my basic facts on plasma membrane that not only plasma membrane is the membrane of the cells, but in case of eukaryotic cells, nucleus, endoplasmic reticulum, Golgi bodies, mitochondria, chloroplast, lysosome, peroxisome and endosomes, all these intracellular organelles are also surrounded by membranes. Not only the plasma membrane is bilayer, but all the intracellular organelles that are surrounded by membranes, they are also bilayer in structure. Now I'm talking about curvature. How curvature occurs? You know that during the budding of virus from the infected cells, curvature can occur. It looks like this. If curvature occurs, then it looks like, let's suppose one virus is actually budding out from the cell, then it looks like this. Okay, so during budding, curvature occurs. So this side is the curvature side of the plasma membrane, this side. Okay, also during the formation of vesicles from the endoplasmic reticulum or from the Golgi bodies, so from the endoplasmic reticulum, vesicles are actually buds out. So these are vesicles that comes out from the endoplasmic reticulum or from Golgi bodies, also make curvature. So how lipid composition controls the curvature? The theory is here that if the head group, we know that the lipid molecules present in the plasma membrane has a polar head group and fatty acid chain, two fatty acid chain. Okay, so lipid contents like phosphoglycerides, sphingolipids, they contain a polar head group and two fatty acid chain. If the head group is large, those lipids contain head group that is large, and if the fatty acid is long enough, then it looks very similar like a cylinder. The lipids looks like cylinder. Okay, remember this, cylinder. Like phosphatidylcholine and phosphatidylserine, these two phosphoglycerides have large polar head groups, and if it contains long fatty acids, then these two lipid molecules looks like cylinder. And those lipid molecules like phosphoglycerides, example is phosphatidylethanolamine. Phosphatidylethanolamine contains a smaller polar head groups, but let's suppose the fatty acid size is same. Okay, then how it looks? It looks like a cone shape. It looks like a cone shape, not a cylinder shape. It looks like a cone shape. So I can write here cone. Example is PE. Here example is PC or PS. Phosphatidylcholine or phosphatidylserine. And here PE, phosphatidylethanolamine, has a small polar head group. If the lipid contains small polar head groups and the length of the fatty acid is same, then it makes PS makes a cone shape lipid molecules. Now if we zoom this curvature area, we will find that, let's suppose if I zoom this curvature area, let's suppose this is the bilayer structure. Okay, it has been found in the curvature area, these cone shaped PE molecules are actually more, they present more here. Cone shaped lipid molecules are present inner side of the membrane, inner lipid membrane. Okay, and the outside contain, let's suppose the outside contain the cylindrical shape of the lipid molecules. Also cone shaped lipid molecules can be present, but more cylindrical lipid molecules are present outside. So when the inner side of the membrane contains cone shape, sorry, here it will be PE, cone shape lipid molecules, it form curvature. It is the tendency. Now if we zoom the flat area, the flat area, then the bilayer will contain more of the cylindrical lipid molecules. So the cylindrical lipid molecules makes, obviously cone shaped can be present both layer, but the number of cylindrical lipids, if it is high, then it make the flat plasma membrane. So cylindrical lipid molecules will make flat membrane, whereas the cone cylindrical, the spelling is wrong, cylindrical. Now cone shaped lipid molecules will make, if it is inside inner layer or the cytosolic layer, then it will make a curvature. So you can see that how lipid composition controls the curvature of the plasma membrane also. So curvature occurs during virus budding or vesicle formations from endoplasmic reticulum or Golgi complex. So this is the curvature. Now let's talk about how lipid composition controls the stability of the plasma membrane. Let's see. I'm writing the number 8 here under the stability. Okay, so cells can be in two kinds of conditions. One type of cells that are subjected to less stress. Let's suppose one kind of cells, so cells that are subjected to less stress conditions in the body. Scientists have measured the chemical composition of these plasma membrane lipids of the cells that are subjected to less stress. They found that the ratio of they found that the ratio of phosphoglycerides, I'm just writing here phosphoglycerides, and cholesterol and the sphingolipids. So when scientists calculate the ratio of sphingolipids is to phosphoglycerides is to cholesterol, they found that in case of cells that are subjected to less stress, the ratio is here 0.5 is to 1.5 is to 1. So that means here phosphoglycerides lipids are higher in percentage than the sphingolipids. But cells that are cells subjected to stress conditions. Now what is the stress conditions? Like epithelial cells that line the gut or stomach, because in the stomach the foods are actually digested, so the pH of the stomach is very low, and the acidic nature of this lining actually creates the harsh environment. So thus epithelial cells that actually line the gut or the stomach, they actually face the harsh environment. Now one thing we have to remember, that epithelial cells that lines the gut or stomach are actually polarized cells, called polarized cells. Let's suppose this is the nucleus and this is the basal lamina. Why this is called polarized cells? Because these cells contain three distinct domains. The outer domain that face the gut or the lumens of the stomach, this is called apical portion. Apical. This portion is called lateral, and this portion, the down portion of the membrane. So this is the membrane. I am talking about membrane. Let's suppose this membrane is the apical, this membrane is the lateral, and this membrane is called basal because it faces the basal lamina. So epithelial cells that lines the gut and stomach are actually polarized cells. Those cells contain this distinct type of plasma membranes. They are called polarized cells. So polarized cells, the apical plasma membrane face the harsh environment, and cells that are subjected to stress, specifically the apical plasma membrane, the ratio of sphingolipids is to phosphoglycerate to cholesterol, if you measure, you will find 1:1:1. Now here the phosphoglycerate percentage decreases, but sphingolipid composition increases. Why? Because I have already mentioned that sphingolipids is more optimally stabilized because of the presence of more long and saturated fatty acids. So you see how the lipid compositions can also control the stability of the plasma membrane.